Well, good evening, everybody, and thanks for taking time out on this beautiful day, beautiful September day, to learn something about climate change and lobsters. And I'm really excited to share this with you. I know that some of you may have heard some pieces of this talk before, but I have changed things a little bit. This is a long-term project, so there's always updates and things to let you know about. So for some of you, this might be new, and for others, this might be sort of an update of what's going on in our Gulf of Maine. And I'm going to touch on some other things as well. And as Suzanne said, I'm going to try to take some pauses and let you either come off of your mute and ask a question, or type something in the chat box if you want as well. So let me just begin by saying that I don't do this work solo by any means. This is most large research projects are done in tandem with people from lots of different places, sometimes different walks of life, and lots of different institutions and partners. And we're just so lucky to have those kinds of people associated with this project and with our Wells Reserve. So I just have to pay tribute to those folks who are pictured. This is the core group of people involved in this project, and you'll learn more about them throughout the talk, but there's certainly many more folks, and I will highlight some of them as we go through this talk. So I'd like to begin, I would like to begin by having you actually use your chat box if you want to. And I'd like you to just take a minute. I'm going to pause for a minute right here and ask you to sort of address this question that I'm throwing out to you, which is, when you hear the words lobster and climate change, what comes to mind? So, whatever it is, please write that down or share that in the chat. I'd love to hear more about your thoughts on that. So I'm gonna stop talking for a minute. I'll actually put myself on mute and ask you to maybe write something down or type something in. Okay, this is a savvy bunch of people. I think there's a lot of people that have been reading and know about this topic. And these are all really good thoughts, comments, and observations. And they are, for the most part, all correct. So I'm really impressed. And so just to sort of capture what these themes are, we've got warming waters, lobsters moving, changing their distribution. We have impacts to the Maine economy, and we have changes to the ecosystem. And so you're all correct, and these are all really important important pieces to this larger puzzle that we're talking about, which is lobster. So I'm gonna try to sort of address many of the things that you have talked about or you've commented on in the chat box. So this is great, and at least from a selfish point of view, now I know who my audience is. So I only have one talk though, so can't do more than that. So let me segue to estuaries for just a minute. And estuaries are, as we know, as most of you know, are really, really important ecosystems all over the world. They are really important in Maine, and we happen to be located, the Wells Reserve at Lawn Home is located on an estuary. We are in proximity to two separate estuaries. And what's interesting about these estuaries is no matter what we study in those estuaries, they are intimately linked to the greater Gulf of Maine. And so we always have to sort of remember that, because we have lots and lots of river systems. The Gulf of Maine is a very large system. It's over 180,000 square kilometers in size. It has some of the largest river systems in the country. With all the precipitation and rain that we've had this summer, the second half of the summer, we've really been inundated by a lot of fresh water in our system. And you don't have to look too far than going outside to any creek, river, tributary, and see how sort of bloated these systems are right now. But the Gulf of Maine is really influenced by our estuarine systems. It's influenced by large-scale currents, small-scale currents, but our estuaries are really linked to the Gulf of Maine. And that puts us in a really unique situation as a reserve when we initiate all our monitoring that we do. So as some of you know, we have a very large water quality marsh habitat monitoring program, and we integrate lots of different other monitoring initiatives into that. And the one area that sometimes we overlook are organisms, monitoring organisms, learning more about how organisms, in particular lobsters, how they respond to change. And so even if we're tasked with the initiative of identifying short-term variability and long-term change in our ecosystems, whether that's water quality or changes in vegetation in our salt marshes or nutrient sampling, we also have to look at how our native organisms like lobster are responding to change, both short-term and long-term. So keep that in mind as I go through this talk here, because it's a really important attribute to what we do. As I know that many of you are already aware, climate change in the Gulf of Maine is been pretty dramatic over the last few years. And I know that these articles are a little bit on the old side now, but they still sort of underscore the chronic issue that we have. And that is that the Gulf of Maine is warming at a very alarmingly high rate. It is changing very quickly. We have an influx of invasive species. We have range expansions by other species. We have all these pressures in the Gulf of Maine that we didn't have 20, 30 years ago, and many of which are really not well understood even today. And so we're really trying to get at that through monitoring and research programs that we have. But in general, I think I want to focus on temperature here, because that seems to be a very significant impact on pretty much everything in the Gulf of Maine. And some of you have seen this type of graph before. This is from a paper in the journal Science, published back in 2015 by Andy Pershing at the time. And again, you see this warming trend. You see this warming trend over the last 30, 40 years. But what's really interesting is that rate of change. So if you look at that we call this the hockey stick model. And if you look at that red diagonal portion of that plot, what you'll notice is the slope of that change is really abrupt. That's an anomaly change. So it's not the absolute temperature change, it's just how much of a change have we experienced in the last 15 to 20 years. It's quite a bit. And lots of other papers and research have sort of accentuated and underscored that as well. So when you look at lobsters and how they fit into this whole paradigm of climate change in the Gulf of Maine, you have to understand with this little cartoon that I've made up that lobsters have a complex life cycle, and they have a larval stage where they've got 4 different stages of larvae that are floating around like plankton in the water. They have eggs. They have eggs that are clung to the underside of their abdomen that can take up to a year to develop. Really, really long time. Once those larvae settle to the bottom and become what we call a benthic lobster or an early juvenile lobster, it can take years, somewhere in the order of 7 years for that lobster to mature to the size that you would find, in your local, supermarket or restaurant. So every single stage, every single component of this life cycle, and I'll talk more about it throughout this talk, is influenced by temperature, as well as other things. But temperature is really what we call the sort of the first order, or the the most prominent sort of catalyst to all these other portions, all these other pieces of their life cycle. So it's a really important thing to keep in mind as well. I'm going to pause for a minute and see if anybody has any questions up to this point about climate change in general in our Gulf of Maine, or anything about lobster life cycles. And Suzanne, maybe you can read me, if there's any chat questions, feel free to just read those off to me. My multitasking skills these days are a little slower.
I don't see any yet.
Okay, then I'll just keep going. But I show this life cycle because it's an important piece to the rest of what I'm going to be talking about. Okay, let's dig a little bit deeper into this climate change, or what I would like to call ocean climate change in the Gulf of Maine. I really want to drill down a little bit into what exactly is going on in our Gulf of Maine. It's warming. We've already talked about that. This is a plot. This is a graph on left side of your screen that's looking at the sea surface anomaly over from 1985 to 2012. And so there's lots of areas that are warming faster than they should be. That's sort of the take home. Some other sort of summary pieces about the Gulf of Maine changing. There's alterations in the type of what we call suitable habitat. And what I mean by that is, and somebody had mentioned it in the chat box, is lobsters having less of a thermal environment. So in other words, thermally favorable temperatures. Temperatures that they like are now becoming temperatures that they don't like. And so they may be moving further offshore, not necessarily further north, by the way, but further offshore to gain more stable, cooler water. They also, in some cases, the thermal habitat is expanding for the better. And that's kind of a counterintuitive to what you might think, because if some areas are warming in the Gulf of Maine faster than others, that actually might help larval lobsters, those pelagic larval stages, get out of the plankton faster. It might actually help them with their development. But the long term, we see that, or as well as other people see that as a decline in their survivorship. So it's sort of a catch-22 there in terms of what we call thermal habitat. And then a paper came out a couple of years ago about changing phenology. And if you don't know what phenology is, it's the idea that seasonal patterns, like the times that leaves should be changing color on a tree. Does it happen earlier one year or later another year? There's factors that influence those phenological patterns. Same thing is true with crustaceans like lobsters. They may change their distributions based on how fast or how slow different portions of their environment are warming or cooling. And so that is changing as well. Those patterns might be happening sooner Sometimes they're happening later. Sometimes they're expanding and happening for too long a period of time. So these are all things that we try to take in consideration when we're sort of constructing all the pieces of a lobster's life history. There's a paper that I've read. It's one of my favorite papers, lobster papers recently, talking about these expanded and contracting thermal habitats. And at the bottom line of this, the bottom line for this paper is that the authors state that we really need to start managing our fisheries, our commercial fisheries, in a climate-ready manner. And that's a whole other talk in and of itself, other than to say that we need to address these things directly, and we need to have management plans that are going to be sensitive to those changes. And I think that's something that we're seeing now, because in southern New England, where lobsters are really not doing well, harvesters and fishermen are changing to other species, but these other species haven't been well, are not even managed very well, because we don't know anything about them. So this is something that we're going to want to keep an eye on, and we're going to want to make sure that we address more squarely, especially people who are in fisheries management. That's an important attribute. This is just another graphic I'll throw up here just for a minute. This is showing the changes in lobster catch from the 1970s to the mid 2000s. And you can see it's pretty easy to see that those purple blotches are moving further north. And so we are seeing, in some ways, a northward expansion, but we're also seeing an offshore or eastward expansion. And so, again, these distributions are changing. We have other issues in the Gulf of Maine as well. I won't get into this too much, other than to say that ocean acidification is a problem in coastal waters as well. The idea that we have more carbon dioxide in our atmosphere, the best place to put that carbon dioxide, the best place for that to dissolve is the ocean, because we have more ocean than we do land. And
Jason, it looks like you froze for us. Oh, okay. Sorry, folks. I think Jason's going to probably log back on. I wasn't sure if it was just on my end or everybody's. Thanks, Mary, for putting that in the chat. We'll just wait a minute. Here he is.
well, that was interesting.
Hey, Jason, you froze for us. What happened on your end?
I wish I could tell you. Let me see if I can pick up where I left off.
Great.
I'm sure that's never happened ever on any talk, because Zoom is perfect. Sorry about that, everybody. Any questions so far? Since that was kind of a easy pause right there.
There's nothing in the chat, but
all right.
Yeah.
So I think where I left off basically was that ocean acidification is a problem in our Gulf of Maine, and that is also affecting lobsters. So they're not just getting hit by warm temperatures they're also getting hit by changes in our ocean chemistry. So, and I want to show you an example of the issues that we're having in southern New England as a sort of a example by which things, may be the same in the Gulf of Maine at some point. So if you look at this map, this is a map of Buzzards Bay in southeastern Massachusetts. There's Martha's Vineyard there. And the Massachusetts Division of Marine Fisheries has been doing surveys on the distribution of egg-bearing lobsters. So those lobsters that hold on to those eggs, right, they're walking around, they undergo these seasonal migrations. And what they've seen over the course of a decade or so is that egg-bearing lobsters are moving further offshore. So the red and the purple, the red are from the 1990s, and the yellow dots are egg-bearing lobsters from about 10, 15 years ago. So you can see that the presence and the distribution of those lobsters is changing. And I want to point out that that's not changing. They're not moving further north. They are moving further southeast, really. And they're moving into deeper water. That's really the take-home message here. And so when we started noticing this and working with colleagues in southern New England a while ago, we kind of came up with this idea of, well, I wonder what that would look like in the Gulf of Maine. Is this going to be the same thing that happens in our area? And it's a really valid question, right? Because if these lobsters are moving further offshore, they're presumably hatching their eggs further away from land, and we want to know what happens to those larvae. So this is really the motivation for why we're doing some of the studies we're doing right now. And then we sort of superimposed that idea in our head with some fine scale tracking data that we've done over the years. So these are lobsters that we've tagged in places like Portsmouth, New Hampshire, and off the Isles of Shoals in Maine, and put little backpacks on their back, and did this with egg-bearing females to see where these lobsters go. And they do tend to go offshore. They tend to go in the summertime. In the fall, they move offshore and they hatch their eggs somewhere out here. And what we're wondering is, will they have to move further someday? Will they have to go the extra mile, as they say? We don't know the answer to that yet, but that is sort of where we're heading. So what I'm going to do for the rest of the talk here is talk about sort of three separate initiatives that we're doing to kind of answer that big, broad question, which is, how are egg-bearing lobsters going to be impacted by climate change in the Gulf of Maine? So the first thing we're trying to do is assess how sensitive are these egg-bearing lobsters, and fishermen call these eggers, right? That's the same thing. So I tend to call them eggers as well. How these eggers, how these moms are going to respond to differences in temperature. The second thing is how those larvae are going to be able to, how are they going to do in terms of their swimming ability to get back to shallow coastal areas where they settle. And the third thing is using sort of oceanography and these drifters that you see pictured, these underwater drifters, to sort of help us visualize where larvae might go based on the movements of those females. So it's sort of a three-pronged idea here, and we're using both lab studies and field-based studies to answer those questions. So the first part, just so we're all on the same page here, we're trying to assess these thermal preferences. We spent a good part of COVID, the lockdown part of COVID, building this giant tank, this giant aquarium tank that you see. It's got all kinds of wires coming out of it, and air bubblers, and you see all these baffles in there as well. So what it's designed to do, it's designed to test a lobster's ability to choose what kind of temperatures it wants. And so you stick a lobster in there, actually you stick two lobsters in there, and you let it decide what it likes. Okay, So there's an example of one lobster that we tagged. You see that little blue thing is a temperature logger. So we know what kinds of temperatures she chose to expose herself to. And that's just an example of some of the raw data that gets read out from that logger. So you can see she's been all over the place. And so our job is to try to take all that data and summarize it for every lobster throughout all the trials we've done. And we've done a lot of trials, and not all eggers are equal, right? So here's this beautiful collage. It's one of my favorite slides because it really shows you that the lobster eggs change color, right? You've probably heard about this. You may have seen lobstermen talk about it. You may have been on lobster boats and seen these yourself. But lobsters go through, the eggers go through different colorations, which are indicative of their development. So the lighter the color, the closer they are to hatching. And so we've been testing egg-bearing lobsters from all different stages to try to understand, do they change where they want to be over the course of their development? And we're really lucky enough to have somebody at the reserve help us actually measure how old these lobster eggs are. And it's been a really cool project so far, and we're really having fun with the data. So I don't have a lot of data to show you on this, and I don't want to overwhelm anybody with statistics and graphs and things like that. So here's what we found. We've run over 150 trials. So we've run over 150 individual lobsters, which is, I'm really proud of our group for doing that because it's a lot of work. And we have found so far that eggers that have late stage eggs prefer colder water. And once those eggs hatch, they tend to move into warmer water. And I'll see if anybody has a reason, if anybody can figure out why they go to warmer water. We have a couple of ideas, but it's an interesting, it's an interesting result so far. And we're really excited to keep summarizing this data. And we have a ways to go, but basically, essentially, on this graph here, you have temperature on the vertical axis and egg stage on the horizontal axis. And you've got, so stage 2 are early eggs and stage 6 are later eggs. And we've even compared small versus large eggers because there's evidence to suggest that there might be a difference there as well. So I just wanted you to get a flavor for the kind of information that we put together. And if this was an in-person thing, I would show you these eggers and I would show you what the data looks like and how we download it and all that stuff. But we'll have to wait for another time to do that. So for now, I'm going to show you this summary. Okay. Then what we needed to do was we needed to have our own source of larvae. So we have these eggers, right? We have these egg-bearing females, but we needed a way to hatch the eggs so that we could grow the larvae, so that we could test the larvae for the other things we want to test, such as how they swim, for example, and how much nutrition they have in warm water. So we were, again, very lucky to be able to have some support from the New England Aquarium and right here at the Wells Reserve, and we renovated some space. We had some space available, we renovated it, and we built a very small little hatchery. So what you're looking at are these orange colored tanks, are these conical tanks where we can put larvae and grow them up into these little baby lobsters. And they're really cute. We don't have any left because we're done for the season for that. But if we do this again next year, we can maybe time it for the summer so that you can come see it if you want. So we basically spent a lot of time growing lobsters, and not an easy thing to do. But we're, again, really happy that we've been able to get larvae. And here's an example of one on my finger, that they're really small, just to give you perspective. And then in the picture next to you, there's a little movie. Hopefully the movie's playing on your end. Is the movie playing? Yeah. No?
Yes,
Yeah.
yes, we
Okay.
can see it playing.
All right, awesome. So this is just a little example of these post larvae. These are about 30 days old, these larvae, swimming around in these tanks. And so the goal here was to produce larvae that we could stick in tanks, change the temperature of these tanks, and swim them for a few days to see how temperature affects their swimming ability. All right, so it'd be like making you do like 100 laps in a pool at a very comfortable temperature and at a very hot temperature. You know, although we're warm-blooded, so we handle that stuff differently than lobsters, which are what we call ectotherms, right? So what we have found so far is that higher temperatures seem to reduce the swimming time. And I'm going to show you another example of that. So here is two tanks side by side, and you can see on the left-hand side are tank number 1. If you can see, they're labeled. Tank number 1 are lobsters swimming at cool temperatures, and lobsters in tank 2 are swimming at warm temperatures. And you can sort of see, and I know you probably have to watch this several times to really figure it out, that the warmer temperatures, the lobsters are stagnating a lot more. And what we actually found is that they change their behavior. So they'll actually exhibit these different kinds of sort of rolling and kind of like free floating. Some of you have heard of the dead man's float, right? Where you just kind of like lay there in the water to conserve energy. So we have been finding that some of these larvae are really just kind of hanging out and floating when they don't like the temperature, when they've got to swim through a really, a temperature that's not conducive for them. That's what we would think of as stressful. So it seems like that warm temperatures are reducing the time that they spend swimming, possibly also compromising their nutritional integrity. So the reserves that they need, the calories that they need for making it from point A to point B. And the punchline that I'll tell you right now is we don't know how far A to B is, by the way. We're also trying to figure that out, but that's a really difficult question to answer. So what we're trying to do is create a proxy for that in the laboratory. Here's what the data looks like so far. So you got a bunch of dots, right, on this plot. You got red dots, which are the warm temperature. That's in red. And the red solid line is the average of all those points. And then you have the blue dots, which are the lobsters at a comfortable 15 degrees Celsius, which is about 59, 60 degrees approximately. And you can see that there is a difference. We haven't run all the statistics on this, so I'm not ready to say that there's an absolute huge difference here, but that there's a significant difference, but the proportion of larvae swimming is much less at warmer temperatures. And we ran these for 72 hours. That was the total duration of these trials. So it's a very interesting result nonetheless, and it's something that we're gonna continue to build on so that we can learn more about especially the behavioral pieces of these swimming trials that we're doing. Okay, are there any questions?
And folks, feel free to take yourself off of mute if you have one. I don't see any in the chat box, but maybe we just wait a second if anyone.
Oh yeah, please interrupt me. This is, we don't, you can come off mute and interrupt
Jason,
me anytime.
Jason, how did you choose those parameters? Is that like 59 degrees or 15 C, like right offshore, or?
Yeah, these are predicted values for, I think, 2050 in the Gulf of Maine. So they come from IPCC data, right? So International Climate Panel reports and predictions that were made for the Gulf of Maine about 50 years from now. Yeah, so that's a great question. Yeah, we didn't just pick these out of a hat. Okay, so let me get to the third piece here. Third piece was really fun, I have to say. We did this during COVID last year, and we did it this year as well. So it was really challenging, but a lot of fun. We got to pretend we were oceanographers for a couple days and build these underwater drifters. So you see that all parts of the drifter are underwater, except for that top portion, which is a GPS unit. The GPS sends a signal to a satellite, tells the drifter, tells us where it is, and we get those pings every 30 minutes. So the idea was set out a bunch of drifters at different locations at varying distances from shore. So we did something like 4 kilometers, 10 kilometers, 15 kilometers, and almost 30 kilometers. And we released these drifter clusters, and we actually did have an oceanographer working with us, so we were sort of legit with that. But we were basically ground truthing the currents in our area. And then we were also looking at using the drifters as a proxy for initial larval dispersal the further you get from shore. So let me show you, the best way to show this to you is show you this little movie. These are drifters that were released on June 24th in 2020. And we let them go all the way until the end of July. So roughly a month, okay, a little bit more than a month. And I'm gonna play it again because it's hard to see this here, but we did release drifters at varying distances from shore. This is from right at the Maine, New Hampshire border. So Portsmouth, New Hampshire, Isles of Shoals, and then past the Isles of Shoals. So if you're familiar a little bit where those areas are, you'll understand that a little bit better. So I'll play this again, and then the map on the right is a static image of where a composite of all those drifters. So without getting into all the nuances of where the drifters went, in general, the drifters that were released further away, only a few of them came back. When I say came back, they came back to the inshore waters. Most of them either went much further northeast, or some of them just went right out past George's Bank and out to the continental shelf. You can see one of them there went all the way down. It looks like it's off Long Island, but it actually went all the way down to Virginia. So at some point we have to shut them off because we have to pay for them. Every time they ping, we have to pay for it. So 5 cents doesn't sound like a lot, but it really adds up. So we were being savvy in terms of the data that we needed for this project. But we learned a lot about the variations in the currents locally, and we learned a lot about if you're hatching further offshore, what that means for larvae. And we repeated this year as well with fewer drifters. I don't have those data to show you right now, but we did get a similar result. So again, we're very excited about trying to integrate that into the other two pieces. So I want to leave some time for questions. So I'm just going to finish up with just a smattering of a few other sort of tangential things that we're doing that kind of complement the project that I've been talking about this evening. These are E-cages. We call them E-cages because they're basically a lobster trap with all these fancy gauges on them. And one of the gauges we put on is a current meter. So the current meter, when it's in the water, that thing stands up like this. And when the current moves, it goes this way, it tilts, it goes 360 degrees, and it measures the current speed and the direction. And so we actually put these cages out where we release the drifters. So we were able to capture current data, salinity data, temperature data, a bunch of different parameters that we were able to capture during the same time that we had where we knew lobsters were hatching in the area and where we had drifters. So this is just one example of the data here in current speed from May through August. And you can see we had 3 different locations. We had a shallow, a midwater, and a deep site. And there are stark differences in those currents as well. Take-home message is deeper water, more stable. Same thing with temperature. If I showed you temperature, it would look similar. Deeper water, colder water, but doesn't fluctuate as much. And so we're learning a lot. And this was sort of a nice little add-on to our project. So that's one very cool thing we're added. The other thing that we added on last year was this lobster contraption that that my postdoctoral fellow, Dr. Ben Gutzler, helped me design. And you can see on the end of that little yellow tether is a baby lobster. He's glued onto the tether. They do perfectly fine. This is like an underwater apparatus that is free floating, has a GPS on the top, a compass, a watch, and a temperature logger. And we essentially just let it go for 20, 30 minutes. And we sort of watch if that larva has any directionality to its swimming. So one thing we don't know in the lobster world, we don't know a lot of things, but one really big thing we don't know is when these larvae are swimming to shore, or when they're swimming in general, we have no idea if they have any preference for the direction they want to go. Is it random, or do they want to go east or west or south? And so this is one way of at least getting some preliminary data on how to do this. So of course, in true fashion, as NASA scientists might do, we gave this an acronym and call it LOIS. So that's something that we've been playing around with, and I'm happy to report that our executive director of the reserve, Paul Dest, actually came out and helped us with this. And I think he had a ball, and it was a lot of fun, and we all learned a lot. So this has been a great add-on to our project. The other thing that we've been using and playing around with quite a bit are these loggers that we stick on the back of lobsters. And this all gets credited to Dr. Ben Gutzler, who is our postdoctoral research fellow, Ford Fellow here at the Wells Reserve. And he has developed these, what we call hat loggers, which essentially are microcomputers that fit inside like a salt and pepper shaker. You waterproof it and you strap it to the back of a lobster, and it will measure heartbeat. It can measure where the lobster is going, what temperatures the lobster is exposed to, light, all kinds of things. And the idea here is that, here's an example run that Ben did in the laboratory. So it's pretty involved, right? You have a lobster in a tank. This is just a sort of ground truthing the sensors here, but you have all these sensors operating in tandem, and he's able to basically watch all the changes that happen in a lobster as it's moving around. So you can gain a lot of information about how a lobster is going to experience changes in temperature, mostly short-term effects, but it has a lot of connections to this larger idea of climate change and how the physiology and behavior of a lobster is going to change over time. So it's a very powerful tool, and it's very it's affordable because you can build them yourself. It's not thousands and thousands of dollars. So some really cool things coming down the pike for us. And I want to end by thanking everybody who's been involved on this project. And I know I may have left some people out. We've got our core project team. We have lots of students and interns, graduate students who are on this project, on some of these projects, and some of our partners as well. So with that, I'm gonna stop talking, hand it back to Suzanne, and I hope I left at least a few minutes for questions, which I'd love to hear from you on your thoughts and anything I might have left out. So thank you very much.
Thanks so much, Jason. I have a question, but I'm gonna hold it to see if anyone else has questions for you first. And feel free to unmute yourself and just ask.
Jason, with the hatch semi, the microcomputer and the drifters, are you able to retrieve that equipment?
Great question, Steve. Yeah, the drifters, we do okay getting them back. The ones that are close to shore, they have contact information on it. So let's see, we've gotten the Coast Guard picking them up. We've had, fishermen and volunteers. Some of them get beached after a while. So we probably get about half of them back. With the tagged lobsters, what Ben ended up doing, and I left this out, is he put a ultrasonic telemetry tag on the lobster. So it's a tag about a little bit bigger than a Tic Tac, glued it to the lobster. That tag emits a high frequency ping. And then what he's done, and I've helped him with this, is we'll go scuba diving where we think the lobster might be. He's got a headphone and a receiver, a hydrophone receiver. And so we'll go swimming around trying to find it. That works about 50% of the time. So he's definitely lost some. Fishermen have pulled them up, and others we have actually found ourselves. So it's kind of a mixed bag in terms of at least this pilot study of having reliability in retrieving them. But that's a great question. Is anybody getting hungry for lobster? Like a lobster roll? I did see that on the chat.
Jason, are researchers predicting where the currents where the currents will shift to, and can you correlate that in the future to what you're doing?
Yeah, that's a really great question. It's probably a better question for a physical oceanographer, which I am not. But suffice it to say, as you might know yourself, the currents are changing. The currents change when temperature changes, when the density of seawater changes, and when you have a higher influx of fresh water. So, when you have ice melt, right, we get water. The Gulf of Maine, one of the sources of the Gulf of Maine's current structure is the Labrador Shelf. So if you're getting fresher water coming in, as well as more fresh water from all our rivers and streams year after year, then you're changing the density of the water, and you may be pushing certain types of water further offshore, or you may be carrying it away, far enough away that those larvae don't have a chance to be where they need to be. And also, I have a colleague, a very close colleague, who's working on changes in food sources that these larvae need. Like lobster larvae are really voracious predators. Even though the larvae are the size of a mosquito, they're really small, but they eat a lot. And we're finding that we don't know a lot about what they eat. We only know that if eggs are going to hatch earlier in the season, that's also going to change. And if there's no food available for those larvae, that will also change the dynamics of what we would refer to as recruitment. So I don't know if I answered all your question, but I think there are model predictions for how the currents might change. The $10,000 question is how we can mesh what we've learned so far with those currents. So we're hoping over the next year, we're going to do some modeling with the oceanography folks and learn a little bit more about, they're really good about currents. We're fairly good about how lobster behavior might change. And so we're going to try to mesh those two things together a little bit better.
Anyone else have a question?
Yeah, thank you, everybody. I appreciate the compliments, but this has been a lot of work by a lot of different people, and I just definitely want to give a shout out to everybody who's been involved so far, because it's been a really good project, and we're still learning and we still have a ways to go, but I think there's some really interesting stuff. And by the way, I want to make one other comment, which is that there's a lot of lobster. There's been an initiative by NOAA in the last few years to basically support lobster research. And so there's several other projects in the Gulf of Maine going on right now, looking at similar things that I've mentioned, and then things that are sort of complementary. And it's really interesting. And I had the privilege of working with some people from Mass Division of Marine Fisheries and down in southern New England, and really saw firsthand how bad the lobster health is down there. It's really not great. And so anything that we can learn from that part of New England and sort of be able to mitigate or advise sooner than later is, I think, going to be, it can't do any harm, I don't think, rather than not continuing to learn, do science, and collect data.
Jason, my question was the same as Steve's about the retrieval of the drifters.
Mm-hmm.
So the half that don't get returned or beach themselves are just floating out there somewhere.
Yeah, yes. And I'm hoping someday maybe somebody from Ireland or the UK will find them, because they tend to get picked up by the Gulf Stream and go that way. I don't know if they'll ever make it. We had some interesting things happen. We had one drifter that was pulled up by a scallop dredger on George's Bank. So they pulled it up at 400 feet. It obviously was destroyed, but we got a FedEx package, and the only thing that was in the package was the GPS unit, which is the size of a cell phone. That was the only thing that was in there. Of course, it didn't work. So, some of them get retrieved even after they've been destroyed. And the drifter materials that we've been using most of them are biodegradable. So they went from vinyl sails to cloth sails. So that cloth breaks down. The mast on the drifter is made of wood. The GPS is not degradable, but yeah, such is life on that one. But we intended, before the pandemic, we 100% intended to have a whole bunch of school kids and college students help us build these things, because that is what they do. This is the best thing you can do, is get students involved and let them learn about oceanography and physics and biology. And we were going to have all these people come here, open up our barn, and have a big drifter build day. And we were really looking forward to that. And then, COVID happened. So we ended up building most of these in our basements. And I think I had my, Ben was in my basement many nights, very late. I'd put food down there and I'd take the plate back and it would be empty. And I gave him a key to the back door. So, you know, a lot of times they'd be like, what's that noise in the basement? Oh yeah, that's Ben. Okay. So we all kind of picked up the slack on that. And there were several of us building these things. It was not the way we wanted to do it, but we didn't want the science. We couldn't put the science on hold for that. And we really wanted to get those out. So we did the next best thing and it worked out really great. And if we do more drifters, I should mention that this year we only released a fraction. We released 24 drifters in 2020. We released 8 drifters this year. Not because we didn't want to release more, but we didn't need to release more. And we did have students, college students from St. Joseph's College in Maine, in the physics class, they designed these little sensors that went on the drifters. So we got half of them back, we got half of the sensors back. And so the students were really, they were glued to their computers and their smartphones, tracking the drifters all over, you know, coastal Maine and Massachusetts. So it was a really fun thing that they did. So we definitely got some student participation in that, for that part of the project.